Hello everyone, in this video I'm going to speak about blotting techniques. Actually, I'm going to continue speaking about blotting techniques, and I'm going to speak specifically about southern blotting and northern blotting. In the previous video, I told you that there are three blotting techniques: western blotting to detect proteins, southern blotting to detect DNA, and northern blotting to detect RNAs. In the previous video, I spoke about western blotting. So in this video, I'm going to speak about southern blotting and northern blotting. Why? Because simply the principle of these two is very similar. So basically, they are the same technique, but with like some differences, because this one is used for DNA and this one is used for RNA. So let's start first speaking about southern blotting technique. As I told you, southern blotting technique is used to detect specific sequence of DNA. So saying that I have a cell, eukaryotic or even prokaryotic cell, and then I extract the genome from this cell, so I have a genome sample. The thing is that I am interested in searching about a specific sequence in this genome, maybe a specific gene or a specific sequence I want to search about in this genome, sorry. What I should do first is that I should perform agarose gel electrophoresis. So step one in southern blotting is agarose gel electrophoresis. I also have there is a previous video in my channel talking about agarose gel electrophoresis. So if you didn't watch it, I also recommend you to go watch the agarose gel electrophoresis video. Now, first of all, the genome or the DNA sequences should be separated using agarose gel electrophoresis. But as I told you before, as the genome is a one long chain, we should first cut the DNA or digest it. And to do so, we use a couple of enzymes called restriction endonuclease enzyme. The restriction endonuclease enzyme is going to cut the DNA in different places and is going to produce several sequences of the DNA of the genome. The thing is that to decide which endonuclease enzyme to use, I should know the sequence I have. Why? Because as I told you in the agarose gel electrophoresis video, the restriction endonuclease enzymes are many. So there are a lot of restriction endonuclease enzymes, and each one of them detect the specific sequence on the genome and cut the DNA downstream this sequence. So in order to choose the restriction enzyme I want to use, I should know the sequence of the gene of my interest. After digesting the DNA or the genome, I apply this sample on the gel, on the agarose gel electrophoresis, using agarose gel and an electrical current. Then the DNAs will be separated. Of course, I might have several samples, and then the DNA sequences will be separated according to their length. The thing is that in order to search about a specific sequence, I should perform southern blotting. Why? Because if I visualize the gel like this, I will see the gel in this way. I told you about visualization in the previous video about agarose gel electrophoresis using ethidium bromide, cybergreen, or other molecules. I would see the gel like this. So the thing is that I cannot differentiate the sample of my interest from other samples, because I'm interested in only one sequence. Sorry, I cannot differentiate the sequence of my interest or the gene of my interest from other genes. And to do so, I should perform southern blotting. Southern blotting, let's say I have this gene, I'm interested in visualizing this gene. So first of all, I should know the sequence of this gene. Let's say this is the gene of my interest. I should know the sequence of this gene, and then I should design a complementary sequence of this gene. So like this, for example, let's say AAGGTCTT, then I should design a complementary sequence. So TTCC AGAA. And then this sequence is going to be called probe. So this probe is going to detect this sequence because it's complementary to it. But how can I perform this? So first of all, in order to perform southern blotting, what I should do is that I should transfer the sequences in the gel or the samples of DNA which are in the gel to a membrane, a nitrocellulose membrane or another type of nylon filter membrane. Why should I do so? Because when I transfer the DNA samples on the membrane, the DNA samples will bind on the surface on the membrane and then the probe can detect the sequence and bind to it, which cannot be done in the gel. Now, how can I perform the transfer step? In order to transform the transfer step, first I should use a chamber like this, filled with the buffer. The buffer is an alkaline buffer. Then I use a sponge. So this is a sponge. Then I put my gel. This is the gel which contains the DNA samples inside. Then I put the membrane on a direct contact with the gel. Then I use paper towels and then I use a weight, a kind of weight, in order to compress all the stuff together. So what I have here is that I have the alkaline buffer, I have the sponge, the gel, the membrane, DNA binding membrane, the paper towels, and then a weight. What's going to happen then in this orientation is that I will have the bottom, which is very rich in water or very rich in the buffer, and then it will in this part there will be high water pressure, and in this part, which is totally dry, there will be low water pressure. Because of this, there will be a kind of capillary forces which is going to drive the water or drive the buffer from the area of high water pressure to the area of low water pressure. So it's going to move the water or the buffer against the gravity from the bottom toward the top. Now what's going to happen is that when the buffer is passing through the gel, it's going to take the DNA samples with. But then when the water is going to pass through the membrane, the DNA samples will stick on the membrane. Why? Because of ion exchange forces. Because the DNA samples are negatively charged and the nylon membrane is somehow positively charged. So there will be ion exchange forces between the DNA samples and the membrane, and then the DNA samples will stick on the membrane. But the water will pass through the membrane because the membrane is a kind of filter. So the water can pass from the membrane and then it will reach the paper towels, which are going to become wet. So I can see that the paper towels are becoming wet, and then I see that the process is working. Now what's going to happen is like this. So this is the gel, this is the membrane, and then when the water is going to pass through the gel to the membrane, the DNA samples are going to be transferred from the gel to the membrane. And then I will have the membrane. What I should do is that I should bake the membrane on 80 degrees of Celsius for 2 hours, or applying ultraviolet radiation to fix the DNA sequences on the membrane. I should perform type of heating, like either using 80 degrees of Celsius or using ultraviolet radiation, like any type of heat. Now what's going to happen is that my DNA samples will be fixed on the membrane. The thing is that these DNA samples are all double-stranded because when I cut the DNA, I cut it as a double strand. So all the DNA sequences which you are seeing here are double-stranded DNA. So if I'm going to apply the probe, which is complementary to the sequence of my interest, the probe cannot bind to this sequence because this sequence is a double strand. So what I should do is that I should treat the membrane with a denaturation buffer. in order to denaturate hydrogen bonds between the two strands of DNA, and then in order to separate the two strands of DNA. So after treating the membrane with a denaturation buffer, all the DNA sequences you're seeing here will be double-stranded DNA. They will be separated. In this stage, I should then apply the probe, which is going to detect the sequence of my interest and bind to it. In order to be able to detect the probe afterward, I should couple the probe with a type of molecule that can give me a signal. This type of molecule can be either a fluorescent, like this fluorescent molecule, or it can be a chemoluminescent. Chemoluminescent is a substrate for a certain enzyme. So if I apply the probe with a coupled with a chemoluminescent, I should then treat the membrane with the enzyme. Then the reaction between this enzyme and this substrate will give me a certain signal. Or I can couple the probe with a chromogenic dye, like any type of dye, which is going to give me also a signal. The thing is that this probe is going to detect this sequence and bind to it. And then I can visualize the membrane in a dark chamber, and then I can see the sequence I have. So I can say, yes, I have this gene in this sample, but I don't have it in this sample. So I can see in which sample I have this gene and in which sample I don't have the gene. So this is southern blotting, sorry. This is southern blotting. Now let's speak about northern blotting. Northern blotting is very similar to southern blotting, but it's used to detect RNA. So let's say I have a cell, this is the genome, these are the RNAs, then I extract the RNAs which contain the sequence of my interest. Let's say that this is the sequence of my interest, and I want to detect this sequence on the gel. First of all, as we did in the southern blotting, what we should do is to apply agarose gel electrophoresis. Now maybe you are asking yourself a question, why would I search for a DNA sequence? So let's say if I want to know if this genome contains a several gene or not, why would I search for DNA sequence? Why don't I search for why would I search for the RNA sequence? Why don't I search for the DNA sequence and that's it? The thing is that why we search for the RNA sequence is to see if this gene is expressed or not, or to what extent this gene is expressed. Because in certain type of cells, the gene might be overexpressed or non-expressed. And to see if this gene, so for example, I performed southern blotting, and I see that I have the gene. Now I see that I have the gene, I know that I have the gene, but I want to know if this gene is being expressed or not. Then I search for the RNA, because if I have RNA, then the gene is being expressed. If I have high amount of RNA for this sequence, now I know that this gene is overexpressed. Or if I have a very little amount of the RNA, then I know that this gene is not so much expressed. Because of this, I search for the RNA sequences. Okay. Now, what I should perform is first. First of all, you should know that the RNA sequences exist always in a secondary structure. So the RNAs are always in a secondary structure. And if I apply the probe, if I design a probe which is complementary to this sequence, and I apply the probe directly on the membrane, the probe cannot bind to this sequence because this sequence is a secondary structure. So what I should do first is to denaturate the secondary structure of the RNA to produce a linear structure of the RNA in order to be able to bind the probe with the RNA. To do so, I apply agarose gel electrophoresis, but with a small addition, which is the addition of formaldehyde to the gel. So what I do is I add formaldehyde to the gel, and formaldehyde will be responsible to denaturate secondary structure of the RNA to a primary structure. which can be then detected by the probe. Let's say that these are the samples separated. Then the secondary structure of the RNA will be denatured to a primary structure, which can be then detected by the probe that I designed for this sequence. Now, similarly to what we saw in southern blotting, let's say this is the sequence of my interest. We should first transfer the RNA sequences from the gel to a membrane, and the process is the same what we saw in southern blotting. So first we use the chamber, we use an alkaline buffer, a sponge, the gel, a membrane. Here we should use RNA binding membrane, which is the same. It's the nitrocellulose membrane, paper towels, and weight. Similarly, the capillary forces are going to drive the water from the area of high water pressure to the area of low water pressure, and then the RNA sequences are going to be to stick on the membrane because of because of an ion exchange forces, because the RNAs are negatively charged, and the membrane, the nylon membrane, the nitrocellulose membrane, is a type of positively charged. So then I will have the RNA samples stuck on the membrane like this. Then I can apply the probe on it. The probe here is an RNA sequence, so we have uracil instead of thymine. And yes, this is the probe. The probe should be also coupled with either with a fluorescent or with a chemoluminescent using an enzyme, or with a chromogenic dye, the same what we used in southern blotting. And then this probe is going to detect the sequence of my interest and is going to bind to it. Then I can visualize this sequence in a dark chamber, and I can see how much RNA I have in this sequence. Then I should know if this gene is being expressed or overexpressed or not expressed. This is everything I wanted to tell you about northern blotting and southern blotting. I hope you enjoyed this video. If you really enjoyed it, don't forget to like, share, subscribe the channel. There are many other interesting videos in the channel. If you have any suggestion for next videos, write in the comments. If you have any questions, also write in the comments. I will answer you, and see you in the next video. Bye.